circular ones that we now refer to as cockle forms. And these are referred to as rods or bacilli. And that's plural. Bacillus would be singular, coccus would be singular. And then we're just going to refer to these, keep these simple over here, as curved forms out of all of that. So these are what we refer to as morphology. Now, I have to kind of warn you, we're going to keep this simple, but there are certain individuals who kind of overlap these and it becomes a problem, but that's typical of bacteriologists. But what we want to consider, and what you're going to be looking for today, are the configurations then associated with these particular morphologies. So what do we mean by that? Well, cocciforms, some species exist as single cocci kind of floating around, and we refer to them, if they have a single cocciform, as unicoccal, or sometimes called monococcal, and that's easy to figure out. So their configuration is that they exist just as one cell, and when they divide, the cells separate and there's still kind of one cell floating around. What you're going to see a lot of today are cocciforms that exist like this. And what would you call those? Good guess, but it's diplo. They're referred to as diplococcal. And then we've got some that exist in, real common on the skin, exist in fours. Hint as to what those might be referred to as? They're tetras, exactly right. So we have tetracoccals. And let me come back up here since we're working down a little lower. And then we have some that exist as little cubes of 8. What might those be called? Okay, and you're wrong. They're called sarsenates. So it's called a sarsenate configuration. And then we got some that occur in chains. And those can be relatively short chains or sometimes very long chains. And those are called streptococcal. And then we've got some that exist as these kind of irregular, the term that's always used in every stinking book that's out there, irregular clusters or grape-like clusters, and those are referred to as staphylococcal. So these are the configurations. The morphology of the cell is what? It's round or cockle. Now these are species-specific characteristics. Species-specific, meaning, how would you define that term?
Each species is different.
Say it again, a little louder.
Each species is different.
Okay, and if
as far as how it looks and
you're on the right track. What I'm getting at here is, does the species typically appear as streptococcal at one point and then some other time staphylococcal? And the answer to that is no. That a given species, genus and species of an organism, typically is going to show this configuration if all the conditions are right. Now, if it's placed in a stressful situation, that might not hold true, but under normal circumstances, that's what the organism is going to look like when you stick it under the scope and take a peek at it. To throw you off a little bit, though, you also know that there is a genus, what? Staphylococcus. There is a genus Streptococcus. And do all of the organisms that show staphylococcal configuration, do they all belong in the genus Staphylococcus? And the answer is no. There are some that belong in another genus, even though they have staphylococcal configuration. So don't get confused by that. And the same is going to be true with strep. Want me to repeat that?
Yeah.
Yeah. I was letting that kind of sink in. So just because a bacterium, you take a look at it, you've isolated it, you've stained it, you're looking at it through the microscope, and it has this configuration, a staphylococcal configuration, does not always mean that it is in the genus Staphylococcus. There are other species that show staphylococcal configuration that are not in the genus Staphylococcus. So just be mindful. The more we talk about this through the semester, you'll get, it's like learning a new language. You get the language down, it begins to make some sense out of all of this. Okay, what about the bacillar forms? A little simpler. So we have a bacillus. It's a rod-shaped bacterium, or at least somewhat rod-shaped. Some of them occur as single cells, hence what would the term be? Configuration is mono or unibacillus. Some of them appear in twos. Pretty common. Diplobacillus. Some of them occur in chains. What would you guess they would be called? Streptobacillus. Those are three of the most common ones. We have some kind of bizarre ones that are referred to as palisades and all of that, but E. coli is one of those that sometimes will fall into that category, but it's kind of a rare situation. So we'll stick with these three because this is by far going to be the most common. And then with these curved forms, we have some like von Leeuwenhoek really kind of drew initially. There are these lazy curved forms, and they're simply called, the best way of defining them is the curved forms. But then we've got some that appear just to kind of be bent, almost like bent bacilli or bent rods. And these are called, typically referred to as the vibrios. V-I-B-R-O-S, a vibrio. There's a whole group of bacteriologists who don't like to put the vibrios in the curved forms. They put them over here in the bacillus and refer to them as the curved rods. And they've got some logic for that because of with genetic studies and things like that, but we're just going to leave them here right now because we're talking about curved forms. But we have to talk about that real quickly here too. What can I use as an example? What am I going to use? I'll use this. If this form is curved like this, yet under the microscope you take a look at it and it looks like this, which would be a rod, and you say, well, how could this be this? Well, if I take that curved form and simply tilt it over, with me? Now all of a sudden you say, well, I've got straight rods under the scope here, and they look like this. But there appear to be some of these in there as well. Well, with time, what you'll do is say, this is the most dominant form. I see some that appear to be rods, and it's simply the way these bacteria are lying on my glass slide. So my perception is going to vary with that. In the same way, over here, if I've got a rod, and the rod gets stuck on the glass slide, then what does it look like? Cocciform, exactly. So just because you find some oddballs on a particular slide doesn't necessarily mean that you have a contaminated culture. Last thing, under the curve forms, is that there is a certain type of bacterium curve form which is tightly coiled. Almost looks like a spring. And this is referred to as a spirochete. Spirochete. Spiral is the prefix on the word. Spirochete. And just a quick comment about these. They're interesting little bacteria. They're really fun to watch under the microscope if you have a living culture. They have a flagellum that comes out of one end and a single flagellum that comes out of the other end. And these flagellum overlap one another and they undulate together. And what it allows the spirochete to do is to corkscrew. So in freshwater, we'll take a look at pond water a little bit later on, we'll find loads of spirochetes. And they kind of zip all over the place. They're non-pathogenic. But there is a whole group of spirochetes that are pathogenic. Namely, Treponema pallidum, you don't have to remember that now, but it's a causative agent for syphilis. And that makes sense because syphilis usually starts what? Breaking the skin in localized infection. Remember, it occurs in 3 stages. Second stage, reproduction, you've been talking about this stuff, right? Been talking about syphilis?
Not yet.
Not yet, we'll get there. Now you know all about it. Second stage is these little guys have corkscrewed out, gone to other parts of the body, and then they erupt on the skin. Finally, third stage is they corkscrew, they bore their way into the central nervous system, into connective tissue, and then you have a problem. But while some are pathogenic, there are loads of others that are not. Okay, this is what we're going to do today. We're going to take a cell from your gum, and on that cell, we will typically find a whole slew of bacteria that have virtually adhered to the cytoplasmic membrane of your gum cells. Now, what kind of tissue? Histologically, what are we talking about here? Found in the mouth, gums, cheeks, it's what?
Simple squamous epithelium.
You said it.
Simple squamous epithelium.
That's not simple. So what's the other option? Stratified. Stratified, excellent. Stratified squamous epithelium. And, you know, what do those look like as you think back? Remember?
Kind of flat like a
Yeah, if you're looking at them from the surface, they are irregularly round, have a really pretty good sized nucleus in the middle of them. So they've got this kind of look to them. From the side, they would look like that. But we're going to take them off of your gum, and we're going to make a wet mount of these cells. Remember how to do that? Yes, no, maybe, kind of, sort of? Let's walk through the procedure. You're going to take a slide, and these are in your top drawer again. Place it on a cover slip. Not on a cover slip. Place it on a paper towel. So you've got this slide now. We want to get a drop of water right in the middle of that slide, and you can get your drop in a variety of ways. You can gently turn on the sink. Where are you going?
Stretching.
Stretch.
And allow it to catch a final drop, or you can do a quick squirt, and usually it'll hang a drop out of that. There he is. So whatever you want to do, we need to get a drop of water, not a teeny-weeny one, so a drop of water right there. Take a toothpick, and you want to use the flat side of that toothpick and get in between your tooth and gum. And we don't want a whole lot of blood here. But you want to do a pretty good scraping on the jawline there. Take that toothpick and stir that into this drop of water. Stir it pretty good. Don't leave the toothpicks lying on the tabletop, and don't share toothpicks. Actually, you know, you don't know if somebody put their toothpick back in this box, do you? Exactly. So once you've got that on there, then you're going to take a cover slip and